Hello students, in the last video we have studied about the female reproductive system and the menstrual cycle. Now in this video we are going to start with the structure of the male gamete and then we'll study the female gamete. Now first of all we'll see the structure of the male gamete or spermatozoa. We should know that in case of male gamete, the occurrence where the male gamete are produced, that is the seminiferous tubule. Inside the seminiferous tubule, there are two types of cells present. One is germinal epithelium and another is the Sertoli cell. From the germinal epithelium, under the influence of the hormone FSH and LH, first of all, the spermatozoa are formed, and when the spermatozoas are formed, then after that they reach to the Sertoli cell. That means the head of the spermatozoa will attach to the Sertoli cell, and later on from the Sertoli cell it get the nourishment. Whenever the spermatozoa completely get formed, then after that and get matured also by the Sertoli cell, then after that it will start migrating towards the epididymis, passing via this intratesticular structure. So first of all we'll see the structure of spermatozoa, how it look like. First of all, occurrence of the structure, the spermatozoa, the formation occurs inside the seminiferous tubule by a process of spermatogenesis. So inside the seminiferous tubule, the formation and development of the spermatozoa occur. Now we'll see that what type of cell is that. First of all, about their measurement. If we see about their measurement, first of all, its length from the head to the tail, it is around 50 to 60 micrometer, and its diameter is around 1 to 5 micrometer. Now this cell, the spermatozoa, is a haploid cell and a motile cell. Motile cell means it can migrate, it can move from the region of the cervix inside the female reproductive tract, and it can reach to the site of fallopian tube. In the fallopian tube we have a region that is known as ampullary ischemic region. In ampullary ischemic region the ovum is present. So the spermatozoa will reach to the site of ampullary ischemic region for the process of fertilization. So obviously that spermatozoa will also have its movement. So in 1 minute its movement is around 3 millimeter in 1 minute. So inside the female reproductive tract, the spermatozoa can migrate around 3 millimeter in 1 minute. So after that we should know its viability. Viability means inside the female reproductive tract up to how much time period it can survive or it can become active. So what happens inside the female reproductive tract, the spermatozoa as it is entering, it can viable up to 2 to 3 days. That means up to 48 to 72 hours it can survive. So its viability is around 48 to 72 hours inside female reproductive tract. After that we'll see that what type of structure is that. First of all, its chromosome number. That means here it is going to be haploid cell. Haploid cell means it is going to have only one chromosome of each set. As we know that in case of human we have diploid chromosome in each cell. We are diploid organism, so that's why in each cell we are going to have twice an chromosome. So from each pair, that means we have 23 pair. From each pair one chromosome will be only present in case of the spermatozoa. So you can see here it is going to have only n number of chromosome. That means we have only 23 chromosome. But here we have a difference. That means the spermatozoa, on the basis of its chromosome number, it can be of two types. That means if you have twice n chromosome in each cell, that means we are going to have 44 autosome and a sex chromosome, a pair of sex chromosome, that is X and Y. So we have a 46 chromosome. Now whenever the spermatozoa is formed from the spermatids, then what happens? From this we will have one set only. That means from the autosome 22 chromosome, and from the sex chromosome, that is from the X and Y, only one of it will come to its spermatozoa. That means either it will have X or it will have Y. That means it is going to have now N chromosome or 23 chromosome. So here on the basis of chromosomes, it is going to be of two types. This one, which is X-bearing chromosome, X-bearing spermatozoa, that is known as gymnosperm, and which is Y-bearing chromosome, that is spermatozoa, that is known as endosperm. So depending upon its chromosome number, it can be of two types. One is gymnosperm and another one is androsperm. So gymnosperm, gynoecium, we already know in the in case of flowers. Androecium and gynoecium we know from that part actually, as it is the X chromosome present in case of female, it is going to have both the chromosomes, they are X chromosome. So we'll use the term gyno here to represent this gymnosperm, and another one is androsperm, which is Y-bearing chromosome. In case of the female gamete or the ovum, in each of the cases, all of the cases, it is going to have only the X chromosome. That means in case of female gamete, all of the female gametes are same. That means it is going to have only X chromosome. So in case of the spermatozoa, if it is X bearing, then after fertilization, it is going to have a pair that is going to have XX. So that's why the baby or that offspring is going to be a female. But if with the female gamete, if that Y-bearing chromosome or that androsperm that is going to fertilize the ovum, then the offspring or that baby will be a male. Now we are going to see the structure of the spermatozoa. Now let us see the structure of the spermatozoa. First of all, the spermatozoa is going to have four parts. So the head or anteriormost region of the spermatozoa is going to have a structure. The tip of the spermatozoa, this is known as acrosome. So first of all, let us see the structure, and after that I will tell you the function of each part. Just behind the acrosome, we are going to have the chromosomes, the nucleus that is present here. So whenever you see the nucleus, this nucleus is elongated, and here posteriorly it is going to have a depression. So that depression is also known as nuclear depression. So here you can see the chromosome number, that is the chromosome number is the haploid cell, and it is going to have only half of the chromosome. So you can see this chromatin fibers, they are compactly present. So this is the nucleus. Now just behind the nucleus, we are going to have centriole. So in case of the sperm, there are two centrioles present. One, the first centriole, which is located in the nuclear depression, this is known as the proximal centriole. We should know that proximal centriole along with the nucleus enter inside the female gamete to help the ovum or that zygote, whenever it is formed after fertilization, that will undergo cleavage or the mitotic division under the function of this proximal centriole. So this is the proximal centriole. Just behind that, we should know that we are also going to have another centriole. That centriole is known as distal centriole. And remember, this distal centriole is present at right angle to the proximal centriole. So this distal centriole have a very important function, and that function is to form the microtubule or that axial filament, which later on is going to give the shape, or that motility will be given by this distal centriole by the formation of this axon name, or also we can call it as the microtubule. So we are going to have this axial filaments. Numbers of axial filaments, they are formed from this distal centriole. You can see here a number of distal centriole are present. Now from this distal centrioles, you can see that from these filaments, only the middle, which is present centrally, only this centrally located axial filament is going to elongate, but which is present on the sides or on the terminal periphery, it is not going to they are not going to be elongated towards the tail region. Remember that from this distal centriole, those microtubules, they are formed from it. Only the centrally present microtubules are going to elongate towards the tail region, but not on the periphery. So those centriole, they're present on the periphery. Those microtubules are not at all going to elongate to form the tail. So you can see here, it is going to form the structure, the tail, that will be given the shape by this distal centriole, by the formation of this axial filament. Now this axial filament will pass through a structure. This is known as ring centriole. Remember, this ring centriole is the structure from where these axial filaments are going to run posteriorly and get elongated towards the tail. But the real function of this ring centriole is still now not understood. Now just see here, we are going to have just the shape. Now the mitochondria, very important thing, that mitochondria are present spirally on this, around the structure of this axial filament. So around the axial filament, this mitochondria is going to elongate, and which is the main structure which is going to give the energy, or that energy, so that the spermatozoa can reach to the site of fertilization. So how it can move, how it get the energy from, it is going to get this energy from this structure, that is mitochondria, and that mitochondria is going to spirally arrange around this microtubule. So this is about the structure. Now we'll see their envelope. So first of all, it is going to have its own cell membrane, but before that we should know that there are another cell membrane. Actually, it is a membrane present around this shape. So first of all, see, around the acrosome and half of the nucleus is covered by one membrane, and that membrane is known as galea. This is known as galea, and just behind that, that is half of the nucleus, and towards the middle piece, it is another membrane present. So this galea will extend. So towards the neck also it is present, and after that it is also present towards the middle piece. So here this shape, this structure, this is known as manchette. So we will see here, this is not the cell membrane. This is another membrane which is present beneath the cell membrane. Above, towards the acrosome, and half of the nucleus is covered by one membrane, that is known as the galea, and just beneath that, this same galea will extend, but we have a term here, this is known as manchette. Now we will see the outermost covering. So here you can see this spermatozoa, this is a cell, obviously it is going to have its own nuclear, this is going to have its own membrane, the plasma membrane. So this plasma membrane ultimately covers the whole structure, the spermatozoa. So this spermatozoa will have its own cell membrane or the plasma membrane towards the tail, passing through this middle piece, neck and the head, and it is reaching to one side. See here, the whole region up to this region the tail ends. That means microtubule or that axial filament will extend from this plasma membrane to a little region. So here in this region of the tail, the plasma membrane or that cell membrane is not going to cover. So this is the plasma membrane or the cell membrane. So this is the structure of the spermatozoa. Now we'll see the region. The spermatozoa is going to have 4 distinct regions. What are they? First of all, it is going to have its head. Then after that, a very small depression is present just after the head. This is known as the neck. And after that, where the nuclear composition, the chromatin is present, this is known as the middle piece. And after that, the structure here, this is known as the tail. But here you can see, whenever you can see that blue membrane, that plasma membrane is elongating, the plasma membrane is not going to cover the whole axial filament. In one region, the axial filament is naked. It is not covered by the plasma membrane. So that structure, that axial filament where that cell membrane is not present, this region up to this very small region where the plasma membrane is not present, that is known as the end piece. So this is about the structure of the spermatozoa. Now we'll see the function of each part. First of all, in case of the spermatozoa, the head. First of all, this head, this acrosome is the main structure who actually penetrate the ovum membrane. Ovum is covered by 3 membranes. So those 3 membranes have to be lysed by the spermatozoa, and then only it is possible that the spermatozoa can actually deposit its nucleus inside the female gamete. So how is it possible? By the acrosome. So that acrosome is actually developed or derived from the Golgi body. And whenever it is developed from the Golgi body, it basically helps in one function, that it releases some enzyme, and these enzymes will help penetrating the egg membrane, that are corona radiata, zona pellucida, and the egg ulema or the vitelline membrane. So what happened? This acrosome, this actually release 3 different type of enzymes. What are they? They release hyaluronidase. This is the first enzyme. Then after that we have the corona penetrating enzyme, and another enzyme that is also released by the acrosome, that is known as the zona lysine. So these 3 enzymes are released from the acrosome, which is developed from the Golgi body, that actually helps in penetrating the egg membrane. Now just come to the nucleus. As you can notice here in the nucleus, I have not drawn the nucleolus. So here in case of the spermatozoa, nucleolus is not present. And why this is happening? Because the spermatozoa, whenever it is developed from the spermatids, the spermatids are non-motile. It cannot move, it cannot swim, and also it is heavier. So in case of the spermatids, whenever it is heavier and non-motile, it is not possible that the spermatozoa can also be non-motile. So first of all, it has to reduce its weight, and whenever it can reduce its weight, then only it is possible that it can swim freely. So that's why, to reduce its weight, that's why a lot of microorganelles starts to disappear from the spermatozoa whenever it was developed from the spermatids. So that nucleus, that cell, actually this is actually a cell, but that cell do not contain the RNAs, as well as it do not have the nucleolus. So nucleolus is absent in the nucleus. And also we should know, here only the acidic proteins are present, but not the basic protein. The basic proteins actually are modified to develop some another structure which are known as protamines. So we should know that nucleus, that is very much important, that nucleus devoid of nucleolus. So this nucleolus is absent in the nucleus, very much important. It also lack the RNA, as well as it also do not have the basic protein. It is also comprised of only the acidic protein. Basic proteins are developed, they are actually modified to some another proteins. They are known as the protamines. Now come to just this structure. See, the nucleus have a depression on the posterior region, that is also known as nuclear depression. On that depression, we are going to have two centrioles. They are known as proximal centriole and distal centriole. What is the function of the proximal centriole? That proximal centriole, whenever the spermatozoa enters inside the ovum to fertilize the ovum, then at that time it's not only the nucleus enters. Along with the nucleus, the proximal centriole also enters inside the ovum. So first of all, nucleus will be go towards the egg region, and after that it will fertilize. Whenever it forms the zygote lack the centriole and it cannot divide. So it requires actually one centriole. So that centriole will be given by the spermatozoa, that is nothing but the proximal centriole. So that proximal centriole enters inside the egg whenever the zygote is formed. After that, to undergo the mitosis process, that mitosis process will be initiated by this proximal centriole. That proximal centriole will form the spindle fiber, and those spindle fiber will separate the chromosomes after its duplication and complete its mitosis process, and from one cell, the zygote is going to contain only one cell. From one cell it can produce two cells, and later on it is going to divide, and we are going to have multiple numbers of cells. Now see the function of the distal centriole. Distal centriole is very much important. Distal centriole will give rise to the microtubule or these filaments. These are known as axial filament, which is going to run posteriorly towards the tail region, and it will also pass through this structure. This is known as ring centriole, but the function of the ring centriole is not understood, or till now it is not understood. So you can see that the axial filament, whenever it is formed, that axial filament, a number of axial filaments are present, but those axial filaments which are present on the periphery, they are not going to elongate. The axial filament which is present only centrally, they are going to elongate towards the tail, and the cell membrane or the plasma membrane will go towards one region, but one part of this axial filament will be still naked. It is not covered by plasma membrane, and it is known as the end piece. Now just see, in the middle piece we are going to have one very important structure, that is mitochondria. I have not labeled it, so let's just see this one. This is the mitochondria. So just see, this mitochondria is the energy source. Energy source means it is going to give the ATP or required energy so that the spermatozoa can swim in the upward direction from the region of the cervix to go to the site of fertilization, that is ampullary ischemic region. So that mitochondria will spirally coil around this axial filament which is formed from this distal centriole. Now this mitochondria have different names or different terms. So in case of the actually we interchangeably use it, but only in case of the insects, the mitochondria have a specific term, and that term is known. This is the mitochondria, which is also known as Nebenkern. Remember, in case of insects, it is present. Now come to the next part, that is the tail part. So this tail part or this end part, this tail region have this axial filament. Whenever you have a cross section, whenever you just cut it and you want to see what is present inside, then that axial filament is going to have this structure which is present in the 9+2 arrangement. That means this microtubules, which is going to give the proper shape to the tail. So that microtubules are present in 9+2 arrangement. That means whenever you see the structure, microtubules are present like this, 9 on the periphery. 2, 3, 4, 5, 6, 7, 8, 9. So 9+2, 9 on the periphery and 2 in the middle. You already know in the botany, we have already studied it. So you can see the centriole, whenever you see the tail region, in the tail region, that microtubule is going to have this 9+2 arrangement. So that is the structure of the spermatozoa. Now after that, let's just see the structure of the ovum. One thing, just remember that this nucleus lack RNA. It is also lack the nucleolus, and the whole cell lack the basic proteins. So these basic proteins are absent. Only it is going to have acidic proteins. So these basic proteins, they are modified. This basic protein is modified to protamines. See, this structure, the spermatozoa have very less cytoplasm. So cytoplasm content is very much less. Now let us see the structure of the ovum. So first of all, we should know the size of the ovum. It is around 0.1 millimeter in shape, and it is very much important to know that it is the largest cell of human body, and this is also visible through naked eye. You should not you can see on by naked eye also. You cannot if you don't use any microscope, then also you can able to see it. It's visible through the naked eye. Now to see this ovum, this is formed where actually the ovum is formed. See, in the menstrual cycle already we have studied that the ovum is formed in the ovary, and it is undergoing a process, a step, that is the process of oogenesis. And by the process of ovulation in the mid of the menstrual cycle, it is popped outside this ovary and it is catched by the fallopian tube. So that structure is known as the secondary oocyte. So in case of secondary oocyte, if you see the secondary oocyte is going to have the membranes, the outermost membrane, that is known as the theca externa. The outermost membrane is known as the theca externa. Just behind the theca externa, we have another membrane, and this is known as the theca interna. So outermost membrane is theca externa, and inside we have another membrane that is known as the theca interna. So another membrane we have here, this is theca interna. we should know this theca externa as well as theca interna, both of them, they are the epithelial tissue. So in case of epithelial tissue, we know that it is lined by a basement membrane. So basement membrane is present just behind the theca interna. So this blue colored layer, this is the theca interna. So here, just behind the theca interna, we have a membrane which is non-cellular. This is the basement membrane. So basement membrane is present here, just behind the theca interna. So this is basement membrane. Now inside the basement membrane, here we have a cavity. That cavity is known as the antrum, and that is going to have its liquid. So this liquid is known as liquor follicule. Then also here we are going to have numbers of cells of granulosa. So numbers of granulosa cells are present here throughout this shape, and also here inside, actually covered by this granulosa cells, we are going to have the secondary oocyte. So here this is the secondary oocyte, which is going to have its own membrane, and outside it is going to have another layer, and that layer is non-cellular layer. So this non-cellular layer, this is known as the zona pellucida. So zona pellucida is formed even inside the ovary whenever it is in the state of Graafian follicle. So here you can see these are the cells, this blue colored cells, they are known as the granulosa cells, or here this one we can also call it as the cumulus oophorus. So these are cumulus oophorus cells, layers of cells are present. Now inside here we are going to have this egg. Actually this is not completing the second meiosis, so that's why this is known as the secondary oocyte. So covering this secondary oocyte, we are going to have a membrane which is non-cellular layer. This is known as the zona pellucida. Now here this zona pellucida, this is actually by a stalk. Here this egg is present in this exact location by a stalk. This is known as discus prodigenum, which is not at all, here it is not important. So discus prodigenum is actually one stalk like structure by which this egg or this secondary oocyte can be attached in this exact location. It will not going to change this location. So here this cavity is developed here, and that cavity is known as the antrum. So this antrum contains the liquid, and that liquid is known as the liquor follicule. Now this cell whenever undergo ovulation, so this is actually the Graafian follicle. This shape, this structure we have drawn here, this is known as the Graafian follicle. So in the state of follicular phase, we can see that the ovum is formed in the preparatory phase. Graafian follicle is formed in the preparatory phase even. So what happened that this Graafian follicle undergo ovulation, and during this ovulation, this cell, this secondary oocyte undergo the first meiosis, and after that it is going to form two cells. One is the first polar body, and the another is not, here this is the primary oocyte itself. So this primary oocyte, first of all the primary oocyte undergo first meiosis, and then it will form the secondary oocyte. So it undergo, this Graafian follicle will undergo first meiosis, and only after first meiosis it forms two cells. One is the first polar body, and another is the secondary oocyte. Till now it is not secondary oocyte, so here the secondary oocyte will be formed after ovulation. So whenever ovulation completes, the secondary oocyte will reach towards the site of fertilization. That means it reaches to the site of ampullary ischemic region of the oviduct or fallopian tube. So whenever the secondary oocyte will reach to the fallopian tube, then this structure here, some of the cumulus ophorus cell or granulosa cell along with the zona pellucida, it comes outside. So here we are going to have the outermost cell, layer of cell. These are the cumulus ophorus cell, and whenever this cumulus ophorus cell are present, along with that we are also going to have another some cells. So these cells will radiate from the middle. There are actually some loosely packed cell, and those cells are known as the corona radiata, as it is radiating towards the outside. So that's why it is known as the corona radiata cell. 2 or 3 layers of the corona radiata cells are present here. This is the corona radiata. And outside here, these are the ophorus cells. Now this ophorus cells and corona radiata cells, both these layers, 2 or 3 layers, that can be stick to each other by a layer, their sticky substance. This is mucopolysaccharide. In this case, this is hyaluronic acid. So because of hyaluronic acid, this ophorus cell and corona radiata cells can be attached to each other. Whenever the spermatozoa will start to penetrate the ovum, at that time, whenever this spermatozoa will penetrate, at that time the acrosome will release the hyaluronidase, one enzyme I told just earlier some time. So that hyaluronidase enzyme will break actually this corona radiata cells and the ophorus cell, and because of that the corona radiata cell will start to disperse from each other, and because of that only the spermatozoa can enter inside the ovum. So here these are the cell, cumulus ophorus cells and the corona radiata cells. Both of them, they can be glued to each other, attached to each other by the mucopolysaccharide, or we can call it as the hyaluronic acid. Now inside we are going to have the same cell layer, that is zona pellucida. So zona pellucida is still present here. Zona pellucida along with the secondary oocyte, whenever it is formed, zona pellucida will also enters inside it. So this is the zona pellucida. So one thing you should know, that the zona pellucida is a non-cellular layer. It is made up of glycoprotein but not made up of any cell. One thing you should know, that the zona pellucida is very much important in terms of fertilization. Why? It helps in species specific recognition. That means inside the female reproductive tract, if the spermatozoa of any other species, not human, any another species enters inside and try to fertilize the ovum, it will be not allowed. And why it is not allowed? Because the zona pellucida readily can recognize whether it is the human sperm or the sperm of another species. So this zona pellucida layer is very much important for the specific recognition. Just inside the zona pellucida, we are going to have the last layer. This is known as the vitelline membrane, also we can call, ulema also we can call, or the cell membrane or plasma membrane also we can call. So this is the last layer, innermost layer of the egg. This is known as the ulema. We can call it as the plasma membrane or cell membrane or vitelline membrane also. Now in between the zona pellucida and the ulema or the vitelline membrane, we have a space, and that space, this space is called as the perivitelline space. So here this is known as perivitelline space. Now inside we are going to have the cell, and that cell maximally contains its cytoplasm. So it is rich in cytoplasm, and this cytoplasm is also very much important because towards the periphery the cytoplasm is very much thick or dense, and the reason is here we have some granules. The cortical region is going to have some granule, so this is known as the thick cytoplasm. So whenever fertilization process will go on, then we'll see that the cytoplasm will block the polyspermy so that more than one sperm cannot fertilize the ovum. That's why we have a reaction that is known as cortical granule reaction. So that cortical granule reaction will occur by means of this thick cytoplasm. Whenever one spermatozoa or the mature sperm hits or attached to this vitelline membrane, at that time from this region, only in this region, the thick cytoplasm will no more thick, but the another side, from this another region, the layer will be very much harder or thicker, so that the other spermatozoa cannot at all fertilize the ovum. Now inside we will have the thin cytoplasm. Thin cytoplasm, or it is the light cytoplasm. So here this will be the thin cytoplasm. It is pleasant. Here it is also going to have its all other microorganelles. It is going to have the mRNA. mRNA is very much important. This mRNA it is also going to help in the fertilization process. Suppose this is the mRNA. This is going to help the ovum to produce some proteins that is necessary for the process of fertilization. So mRNA will be present, then the mitochondria will be present. Obviously here the nucleus will be also present, but remember this nucleus is present towards a little periphery. It is not present centrally. Exactly in the center it is not present. Nucleus is present towards the periphery, towards the surface a little, and that type of cell which have the nucleus towards the periphery, exactly not in the center, that type of cell, this is known as the eccentric nucleus. Eccentric nucleus. Why it is called as the eccentric nucleus? Because this nucleus is not central, it is present towards the periphery. Now one thing is important, that whenever this cell, this secondary oocyte undergo the second meiosis, here you can see only first meiosis is completed. So suppose the spermatozoa is about to fertilize the ovum, at that time what happened, the egg or that ovum which is in the secondary oocyte state, it is not completing the second meiosis. At that time what happens, the meiosis 2 is not completed, so it has to first of all complete the meiosis 2. So as soon as the spermatozoa will attach to the egg site, metaphase promoting factor will be get activated, and because of that the ovum will undergo second meiosis, and whenever second meiosis completes, it produces once again 2 cells, and those cells, they are the ovum and the second polar body. So here you can see a cell or that cell is not at all going to help in fertilization, it will be removed outside. So that cell, this is known as the first, this is the second polar body. So this structure will be the second polar body after second meiosis. So when second meiosis is completed, we will have 2 cells. One cell is the ovum and another is the second polar body. Now one thing you should know, that here we will develop polarity. Polarity, why? What is the polarity actually? Whenever this whole ovum is actually surrounded by a number of spermatozoa, then at that time the ovum will complete the second meiosis. So whenever second meiosis is completed, at that time from the cell the second polar body will be removed, and whenever the second polar body is removed, That region from where the second polar body is removed, that region is known as animal pole. Remember, polarity is developed, so this region from where the second polar body is removed, that is known as the animal pole. And exactly on the another region, exactly the opposite region of the animal pole, this region will be known as the vegetal pole. So here animal pole and vegetal pole will be developed inside the egg. Actually, whenever second meiosis is completed, second polar body will be removed from it, and whenever the second polar body is removed from that region from where the second polar body is removed, that region is known as animal pole. And opposite to the animal pole, we will have the vegetal pole. Now, here you should know that whenever second polar body is removed from that egg, in that region, this vitelline membrane, zona, pellucida, coronary data cell will be little bit dispersed, or in this region, that cell layer is not present. So, better that spermatozoa from that region will fertilize the ovum. So, that region, the animal pole region, is the site where the spermatozoa will attach to the ovum membrane or this vitelline membrane, so that it can fertilize the egg. Another important point is, whenever fertilization is completed, we will have a twice-end zygote, and that zygote, one cell zygote, that will undergo the process of duplication. That means from one cell it will produce two cells. So two cells will be formed by a process of mitosis. So mitosis will occur by forming two cells. So how this two cell will be formed from one cell? By undergoing cleavage. So cleavage will also occur from the animal pole vegetal pole axis. So whenever we'll have this animal pole vegetal pole axis, from that region one cell will divide to form two cells, and each cell will be known as blastomere. So this is the human egg. One thing you should know, that egg contain the yolk. In case of human egg, the egg do not contain egg yolk, or that yolk is very much less. So that type of yolk which is not present, we can call it as the alacetal egg. So in case of human egg, this is alacetal, or here it is not going to have the yolk. In case of some other organisms, like in case of birds, in case of reptiles, in case of those organisms, the yolk content is very much high. As we can see in the chicken egg, that contain a huge amount of yolk, so that the growing embryo can get the nourishment from the yolk. And that yolk is formed from a specific chromosome, and that chromosome is the lamp brush chromosome. So lamp brush chromosome is present in case of reptile, or in case of apes or birds, which can develop the yolk. But in case of human, it is not at all present. In case of human egg, it is also not covered by any type of shell, and that's why this shell or that calcareous shell is not present. Why it is not present? Because human is viviparous. In case of human, the egg not at all come outside the body. This remain inside the fallopian tube. It will be fertilized by the spermatozoa, and after that zygote will be formed, and that zygote will undergo multiplication, and embryonic process will be completed within the uterus. So in any case, the egg will not at all come outside the body. So that's why it will not at all require any type of protection. So that's why the outer hard shell, calcareous shell, is not at all present. So this is about the ovum. This is the structure. In the next video, we'll see the process of fertilization. Then we will see implantation. Later on, we'll see the whole process of embryogenesis. Thank you.